Well, morning, everybody. Welcome back to Bio 1120. What's the guy doing in the lower right here? Is he like putting like a plaster heart on that wall? Is that what's going on, or is it like a patch? I'm just puzzling about that myself. A student organization asked me to put this up for you before the start of class today. I said that I would. I have. There you go. All right. I know Katie. She was a student in this class like 3 years ago now. It's just swell, right? I'm sure it's an interesting group, and check them out. All right. Here's the work to do today. I want to start this morning just by drawing your attention to one thing, maybe with two parts. Okay? On Thursday, September 29th, so 2.5 weeks from now, when you guys come here, we're going to give you your first test for this class, right? I don't want to be a buzzkill, right? Everybody's happy at the start of class, feeling good, beautiful day outside. But remember, on the 29th, there is going to be a test. We're like about, at the end of the lecture today, I'll be halfway through with the stuff that's going to be on that first test, okay? Here's the thing. Well, a couple things, I suppose. If you are listening to this on Integrity and you're thinking that the normal meeting of the class time isn't going to work for you, you should probably start getting in touch with one of the two supplemental instructors for the course, Lauren Trout in particular, to arrange to see if you can take the test an hour before everybody else does. We do have a room set aside, but there's limited seating, and so it wouldn't be too soon to start thinking about setting that up. For those of you who are here, I don't think that's an issue. Just come back, right? It'll be just like the normal class. Everything will be fine. There are going to be 45 multiple choice questions, right? All multiple choice. You'll need to bring a number 2 pencil because you're going to have to fill in little bubbles on a computer form that will do all the grading for us, right? Trivial stuff there. But do you remember, like, the first 2 times that we've met? I didn't say anything the last time that we met, but the first 2 times that we met, I said the single best way that I can think of to prepare for one of those tests, like the one you'll see on the 29th, is look at some of the old exams that are available for you to look at on the pilot page for the course. I haven't checked to see how many people have taken me up on that suggestion just yet. I think it's largely because I like to avoid things that will disappoint me, all right? So I'm not thinking everybody in the room has done that yet, but I want to shout it out to you. It's not too soon to start looking at those old tests. Look at the old tests available to you on the pilot page of the course. And here's another reason to look at the old tests available to you on the pilot page for the course. It is that if you do that by midnight on Thursday, you do get 1 point of extra credit. That's spelled out in the syllabus for the course. There won't be exceptions, right? If you come and tell me, oh, gee, I didn't think about doing it until Friday. Sorry, the syllabus said 1 point by midnight on Thursday. All you gotta do is look at them. We'll be able to tell. And I think we'll probably send you an email saying, hey, sometime in the next week or so, hey, good job for checking out some of those old tests. You got that 1 point of extra credit. If you didn't, we'll say, hey, you should check out those old tests. Sorry, you're not going to get the 1 point of extra credit at this point, but it's still worth your time. When you look at the old tests, I want you to really look at them at some point. And again, it's not too soon to do that. You'll see from one year to the next, there's a lot of similarity between those tests, right? Students think that I'm lazy, and they tell me, oh, you're just copying the same questions you used in previous years. But I think, you know, out of self-defense, I think that when a student tells me that I'm lazy, that doesn't happen too often, but I know what you're thinking, right? When you think that I'm being lazy and recycling questions, I don't think you've looked at it as hard as you should have, because what I'd like you to do is, especially on those questions that you see from one year to the next, notice how often a different answer is the correct answer. And what I have actually done is I've changed a word, sometimes two words, in the question itself to cause a completely different answer to be the right answer to the question. If you notice that sort of thing, I think that means you're looking at the exams in the right way, and you're thinking about it in the right way. I don't care that you can memorize the right answer, right? Oh, B is the right answer for question 3 every year. That's not important. I want you guys to be able to look at the question, stop and think about it, and then reason your way through it and figure out what the right answer is. I'm testing you on the concepts. I'm trying to test you on the ideas, how this stuff works. And I'll tell you this here, I'll confess to something, a guilty pleasure that I have. After the exam on the 29th, somebody is going to come, not just one, there will be a dozen, maybe 2 dozen, that will come to me and say, gee, Dr. Crane, you know, I looked at the old exam from 2013, and question 3 was the same on that test as it was on the test we took on the 29th. But in 2013, B was the right answer, and the answer key is wrong this year because D is the right answer according to the answer key. How am I going to get my points back? And my guilty pleasure will be the little dialogue that's going on inside of my head where I'm saying, ha ha, yeah, the same question really, let's take a close look at it. It's like, oh, look at that, it's a different question. Huh? Maybe D is right now, whereas B was right before. What I'll actually say is something like, well, gee, the question's actually a little bit different such that D is the right answer, and you know, I have to grade based on what the right answer was for this year's test. Right? So anyway, when you're looking at the old tests, have that in mind. Look for how it is that, and you don't even need to see how I've done it. When you read the question, think, how could this question be changed to make one of the other choices the right answer? And you'll often find that there are ways to do that, because I kind of built that in to the exam questions that I use to test you for this class. So I'm trying to get you to think about the concepts and why it is that something is the right answer, as opposed to simply just knee-jerk, this is the right answer. Okay? So it's not too soon. I accept, I understand that about half of the questions that you see on those old tests are going to be things that, I don't know what he's talking about, I've never seen it. Even those are worthwhile for you to look at, because that'll give you some clues as to what to really key in on when I do talk about those things in the subsequent lectures for the course. Right, so again, not too soon. Lots of them are available to you on the pilot page. If you're having a hard time finding them on the pilot page of the course, I'll tell you, I'll try to help you, but my best help to you would be to point you to the 2 supplemental instructors. They're kind of the ones who are keeping the pilot page all stuck together and cohesive and looking nice. They'll be happy to help you. I'll help you as best as I can. Cool. Let's then get to talking about some biology stuff. I'd like to start really now by reminding you about what's been going on in here. This is a biology class. We're talking about life, living things, what makes us different in some sense from non-living things. We've started at a very reductionist level, some very basic chemistry, subatomic particles, talking about the nature of chemical bonds, electronegativity. Lately, we've been going up the scale a little bit and talking about the chemistry of carbon, as well as the chemistry of water, organic chemistry. And I've been trying, during the last couple of times that I've talked with you, to really encourage you to just stop and look at these structures and know what's going to happen from the structures, how these structures will behave based on your understanding of the nature of chemical bonds. That's pretty much what it comes down to. And today, we're going to take it one step further up the scale. Instead of talking just about atoms and maybe not just functional groups, today I want to start piecing things together and show you some of the cool molecules that living things can make that draw from that painter's palette that I've been alluding to in terms of these different kinds of chemistry, how you might string things together to really make something that can do pretty complicated things. right? One of the things that we talked about in the very first lecture of the class was living things are more organized than their surroundings. Today, when we start talking about biopolymers, I think that's one of the first really clear examples of something that you find in living things, that level of organization that we don't see in non-living things. And when you see those complicated biopolymers, it lets you see, I think, also how it is we can do stuff that non-living things cannot do. So today, I want to talk with you about macromolecules. Ultimately, the plan for me is to talk with you, and this is what's in the syllabus too, by the way, as I'm going to take you by the hand and tell you something about carbohydrates and lipids, and then frankly, very honestly, I'm going to save the best for the Thursday lecture when I'll talk with you about proteins and nucleic acids, right? So we're only going to get to about the lipid part of today. I may tell you a few things about proteins just to save me some time on Thursday, but that's what we're in store for. I just want to ease into it, though, with a reminder of some of the things that we talked about in lecture on Thursday, these functional groups of carbon. So remember this outline from Thursday. Here are the 6 or 7, depending on how you do your counting, functional groups of carbon. And I hope you'll remember, I want to really just keep pounding on this, I hope you'll remember there's some things here that unfortunately you do have to memorize the nomenclature just to make sure that we all are saying the same things and meaning the same things. But really, most importantly, I want to make sure that we're all on the same page about what these functional groups do to the behavior of the molecules that they're associated with. So I'm going to run through these one more time. This is exactly the same stuff that I talked with you about at the end of lecture on Thursday of last week. But here's the deal. 6, maybe 7 different functional groups that we find time and again associated with carbon molecules. And let me make sure, maybe even before I dive into that, why do I say the functional groups associated with carbon molecules as opposed to the functional groups associated with hydrogen molecules or oxygen molecules or nitrogen molecules? Do you know why I'm talking about carbon molecules as opposed to the other 3 big players? What do you think?
Carbon makes up 96%.
The 4 together make up 96% of our mass. I don't know. I think oxygen may be the biggest player. But it's not a mass thing. It's that the carbon of those 4 key atoms, the carbon is the one that you can string together to make some long chains, right? Because it's got a valence of 4. So you can string together long chains of carbon. There's frankly not much you can do with hydrogen because it's got a valence of 1. There's no chain potential there, right? You just tack it on to another thing and it's done. You might be able to do some things by chaining together nitrogens, but if you're interested in making long chains of something, why would you limit yourself to 3 bonds that you can make when you got something in your arsenal that lets you make 4? Right? So that's why we're so fixated on carbon and carbon's chemistry in biology, is because that's the thing that lets you string together long chains and then dress up those chains with these different kinds of functional groups. All right, so first functional group we talked about was a hydroxyl group. All right. That's just the name of this thing. It's a hydroxyl group, okay? When you dress up a carbon chain with a hydroxyl group, we call that molecule now an alcohol, okay? And conceptually now, what I'm really get excited about personally, when you see a carbon chain's got a hydroxyl group attached to it somewhere, I want that to cause you to immediately think, oh, this molecule now is one that will be at least a little bit water soluble. Okay, there will be at least a little bit of a basis for interaction between this molecule and water and other molecules that might be interested in making hydrogen bonds. Okay, so that's first and foremost. When you see a hydroxyl group on any compound you see for the rest of your life, you may be incredulous that this is going to happen, but I'm pretty confident Later in your life, you're going to see carbon molecules, and there will be a hydroxyl group there. In the back of your head, I want you to hear my voice saying, water soluble, a little bit water soluble, right? Because that's what's going to be happening whenever you see one of these hydroxyl groups. I'm sorry that you have to hear my voice as opposed to somebody with more dulcet tones, but that's just the cards that I've been dealt with. You're stuck with my voice saying a little bit of water solubility. All right, so hydroxyl groups. In addition, second functional group, there are these carbonyl groups. I apologize. It's simply a matter of nomenclature. It's the sort of thing you just kind of have to memorize. If you see a carbon with a double bond to an oxygen as part of one of these carbon chains, what you're looking at is a carbonyl group. Carbon compounds with carbonyl groups get called one of two things. They're either aldehydes or ketones. Ketones have the double bonded oxygen in the middle. Aldehydes have it at the end. Okay? Nomenclature, just kind of have to have that same set of words that we're all talking about. That's the deal. But where it gets fun for me, frankly, oh, geez, I got to memorize that. I don't want to memorize that. There must be a way to look that up. But where it gets fun for me is talking then about and thinking then about what that does to the molecule. How have I messed this molecule up? The molecule without that double bonded oxygen was perfectly happy, but I reached in and I changed it, at least in my mind's eye, and I put that double bonded oxygen there. What's going to happen? How is it going to behave differently? It'll behave differently because that's going to be also pretty water soluble, right? Maybe even more so than you would have gotten with the hydroxyl group. And that's the case because there will be a little bit of negative charge associated with the oxygen, a little bit of positive charge with this carbon, and also a little bit of positive charge just sort of diffusely spread out along the rest of whatever's attached over here, because this carbon is pulling electrons toward it because the oxygen has done such a good job of pulling electrons toward it. So things with carbonyl groups have a localized partial negative charge and a kind of diffuse partial positive charge around the rest of the molecule, at least near that bond. Those things make this contribute to the molecule being a little bit water soluble. Now I'll tell you, there's nothing going on with a carboxyl group beyond this being a carbonyl group with a hydroxyl group right on top of it, right? That's exactly what's happened here. A carboxyl group is just the combo meal, where here we have not just the carbonyl group, we also have the hydroxyl group there. But when you dangle a carboxyl group off of a carbon chain, right, that's called now a carboxylic acid. And from playing mind games with the molecule, think about what we've done to this thing. I was going to say completely. We've substantially, maybe radically, certainly very significantly changed the sort of behavior of the carbon compound by dangling a carboxyl group off of it. Carboxyl groups, when you see a carboxyl group, I want you to become conditioned, just indoctrinated, accept that inside of a cell, neutral pH, like you talked about in lab all last week, inside of a cell, that carboxyl group is going to have a full-blown, flaming negative charge associated with it. Okay? A full-blown charge. None of these wimpy partial negative or partial positive charges. This is a raging negative charge. That is going to be chemically reactive. Nature doesn't like that sort of intense, flaming, outlier kind of negative charge. That's going to be chemically reactive. And it's going to be particularly chemically reactive with anything that might have some positive charge to help scratch the itch, to solve the problem that is that intense negative charge. It's also going to be kind of repulsed by other things that have negative charges. So there's an attractive force that's going to be going on to pair it up with something positive. But if you have something else that's got some negative charge, that's going to help keep those two things physically far apart. All right? So carboxyl group. If you put one of those on a carbon compound, we call that carbon compound a carboxylic acid. Frankly, I think only organic chemists, old school ones, call them organic acids. I never hear biologists talking about them. I do sometimes hear biologists talking about carboxylic acids. But the key thing here is that you should expect at normal physiological types of pH, the kind of pH inside of most living things, as a rule, that's going to have a single negative charge. Okay? So very chemically reactive. I hope it goes without saying, very water soluble. So happily, we've got a fourth functional group that sort of gives us the yin to the yang that was a carboxylic acid. Right? So we got something, a functional group. If you want to make something intensely negatively charged, put a carboxyl group on it. If you want to have something that's got a positive charge on it, put an amine group on it. And that's the structures that we're seeing here. An amino group, when you put it on a carbon compound, we call that carbon compound an amine. And at normal sort of neutral pHs, what you would find inside of a typical cell, that amine group is going to pick up a hydrogen and end up causing there to be a local, full-blown, flaming positive charge, right? The perfect match for that full-blown negative charge of a carboxyl group. So very often, I didn't do anything, very often you'll find carboxyl groups and amine groups hanging out together. Huh, interesting. Say no more, right? That's all there is to say about an amine group. That's what we dial in, call up off the bench if we want to have a positive charge on a part of a molecule. This one's kind of odd compared to the other functional groups we've been talking about, right? We've been doing sort of nice gradual walk through the palette of different chemical flavors that you might have. A sulfhydryl group just doesn't follow here. Maybe it should have come up after the hydroxyl groups. Maybe that would have made more sense. That's just not the way people talk about them, though. But a sulfhydryl group is essentially a hydroxyl group where instead of oxygen, you've got sulfur. Okay? Sulfur's valence and oxygens are sufficiently similar that you should expect the sulfhydryl group by itself, when you make a carbon compound into a thiol, you should expect that thiol to behave water-friendly-wise in pretty much the same way as an alcohol would have. So the sulfhydryl group and the hydroxyl group, fairly interchangeable in terms of the impact on water solubility and reactivity. But there is a difference, and the difference between the sulfhydryl group and a hydroxyl group is that sulfhydryl groups, unlike hydroxyl groups, do allow for cross-bridging to take place between molecules. So one sulfhydryl group can rub up against another sulfhydryl group. The two hydrogens of those two sulfhydryl groups can get together and go away as hydrogen gas, and the sulfurs fill their valences by developing a covalent bond between themselves. So where you have one sulfhydryl group on the molecule and another sulfhydryl group elsewhere on the molecule or on a different molecule, you can sort of staple those two parts of the molecule or those two different molecules together by making what's called a disulfide bridge. All right? So you got the two sulfurs and there's a covalent bond between them. That's sort of what makes sulfhydryl groups special. If not for that, the capacity to make those disulfide bonds, I don't think that we would be talking about sulfhydryl groups because they're much less common than hydroxyl groups on the carbon compounds in our bodies. And I don't think nature would have had any use for them either, since sulfhydryl groups are largely doing the same thing otherwise that hydroxyl groups are doing. But that's what makes them special, and I think that's why nature found a need to have this be a fairly commonly occurring functional group for carbon, is that capacity to make those disulfide bonds. All right. And then we're in the home stretch. 1, maybe 2 to go, right? Here's the big boy of negative charge, right? If you thought it was fun to paint with the bold color of a carboxyl group and just, yeah, I'm going to be right in your face, this is negatively charged. Wow, do I have something for you, right? This is the neon pink of the painter's palette, right? It's like, whoa, that's pretty intense. Phosphate groups have a raging negative charge, not just 1, actually 2. Wherever you see a phosphate group attached to a carbon compound, you'd better be thinking, I hope you'll be thinking, oh my gosh, what's going on there? That is as intensely negative a charge as you will ever see. It's going to be hugely chemically reactive. Anything with even a little bit of positive charge is going to feel a strong attraction to it. It's going to be hugely water soluble, right? This is negative charge to the max. You can't see anything in nature that's going to have a more local concentration of negative charge than what you get by putting a phosphate group on a molecule, right? So this is intense negative charge, hugely negative. And I'll tell you just as an aside, editorializing a little bit, I feel a little bit cheated by the fact that we don't have anything that is the yin to the yang of a phosphate group. There is no one functional group that brings to bear 2 positive charges to balance this out, right? With a carboxyl group, I can pair that up perfectly with an amine. For whatever reason, we don't have something with 2 positive charges on our painter's palette, but this is the raging big boy of charge associated with organic compounds. Those phosphate groups bring with them 2 full-blown negative charges. And last, and I think arguably least, are the 7th-ish functional group for these carbon compounds, and that is pretty much just none of the other 6, right? So a methyl group is a carbon atom that doesn't have anything but hydrogens dangling off of it. Okay? If you plopped, let's conduct this thought experiment. If you had something with a phosphate group, if you had a nice carbon chain and at one spot there's a phosphate group on it, okay? What's that going to be like? Ooh, that's going to have a pretty serious negative charge. It's going to be very chemically reactive. It's going to be very water soluble, right? That's the behavior of that molecule. If I swapped out that phosphate group as the functional group and I replaced it with a methyl group, right? How is that molecule's behavior going to change? How will it be different with a methyl group instead of a phosphate group? And that might be a fair exercise to do with all the rest of these functional groups. There's 6 plus methyl groups. If you do that thought experiment, I hope what you'll come up with at the end of this is, gee, instead of the phosphate group, if I've got a methyl group, this molecule, this carbon compound now is not very chemically reactive. It's not water soluble. It's actually hydro-water phobic, afraid. It's afraid of water. It's a hydrophobic molecule. It's going to try to get away from wherever water might be. It doesn't want anything to do with water. Water wants nothing to do with it. It's going to want to coalesce around other like-minded hydrophobic molecules and make a little ball unto itself separate from the rest of the world. All right? A hydrophobic exclusion zone where it's forcing water to go somewhere else because water wants nothing to do with it. So if you put a methyl group on the surface of a protein, for instance, what will that do to the protein? It's going to create a hydrophobic pocket. Let me say that more clearly. It will create a hydrophobic pocket on the surface of the molecule, and it will probably cause that protein to now want to rub up against other things that have corresponding hydrophobic pockets. And especially if you replaced a phosphate group with that methyl group, you can radically change the behavior of that molecule. Whereas with the phosphate group, this is something that the protein's going to want to put up on its surface to get as much access to water and things that are positively charged as possible. If you take that phosphate group off and you put a methyl group on instead, the protein now is going to fold up on that methyl group, try to hide it from the exterior environment. It's going to change the shape of a large portion of that molecule. And we'll talk about this more later, but in biology, structure and function, shape, and the way something works are intimately related. You do that, and you'll almost certainly change the way that that molecule behaves in a biologically relevant way. Okay? So those are the tools. Those are the spectrum of colors that we get to play with. There's a continuum here. There's like a two-dimensional grid with maybe a third dimension. Here are the dimensions to keep your eyes on. One of them is hydrophobicity versus hydrophilicity. Hydrophobicity is how afraid this is of interacting with water, its aversion to water. Hydrophilicity is how much it wants to interact with water. So all of these functional groups fall somewhere on this scale from being completely averse to water to being really comfortable hanging out with water. Another dimension to keep track of here is charge, all the way from flaming negative charge, as extreme as a phosphate group, down to the other end of positive charge, which for us apparently is zeroed out or bottoms out at the +1 positive charge of an amine group, and then several shades of gray in between. One negative charge from a carboxylic acid, a carboxyl group, and then some partial positive and partial negative charges from hydroxyl groups and carbonyl groups, right? And if there is a third dimension, right, so we've got the hydrophobicity, hydrophilicity, and we've got the charge. If there's a third dimension, and this is a small dimension right now, but it'll get bigger on Thursday, it's size, right? So hydroxyl groups are pretty small, relatively speaking. Phosphate groups, relative to a hydroxyl group, pretty big. It's an assemblage of a number of atoms as opposed to just 2 atoms. Right? So the bulkiness is another thing to maybe keep track of. For the functional groups, they're all roughly the same size. Later on, we'll be talking about functional groups that are bigger than that. All right. So that is another way of saying, I think, all the stuff that I talked with you about on Thursday. Same stuff, expressed in a different way, maybe now with a little bit more context. Hopefully, we're all on the same page. That's the deck of cards that we have to work with. 7 cards, 6 for sure. 1 just seems to be the absence of the others. And what can we do with these 6 or 7 functional groups? Well, we can make pretty impressive biomolecules with them. And it turns out that there are 4 commonly encountered biopolymers that show up in all of our cells and in all living things: carbohydrates, lipids, proteins, and nucleic acids. There is no contender for a 5th slot. All living things rely really heavily just on those 4. So once again, nature has done us a good turn, right? Time and again, we see this happening. There's 92 naturally occurring elements. Nature really goes to the well for just 4 of them, over and over and over again, right? There's an infinite, almost, different number of ways you might combine those 4 elements to do stuff. Nature goes to just 6 wells, maybe 7, with these functional groups, right? Did us a good turn there. Instead of having to master an infinite number of combinations, we can just focus our attention on those 6 or 7. And once again, what do you do with these functional groups once you've got them dressing up some carbon chains? Turns out there's probably an almost infinite number of ways you might be able to string them together. Nature doesn't do that. Nature goes time and again just to 4 different biopolymers. It uses those biopolymers for different purposes. Right? That gives us perhaps some interesting insights in terms of what's important for a living thing. What does a living thing need to do to be able to pull off this trick that we call being alive? And keep your eye on that as I talk with you about these 4 polymers, because apparently that's pretty much the whole story. At the heart of it, that's what it takes to pull off these tricks, what these 4 biomolecules do. So carbohydrates, lipids, proteins, and nucleic acids. Let's go by one by one through that list and talk about what makes them special right after I talk with you about how it is that they get strung together. And so that's what this polymer stuff is all about. And I tell you, as I was thinking about this morning, looking at the slides before lecture, I was thinking, I got the sequence here backwards. I should have this slide come at the end. Let's talk about this slide first, then the next slide, and then go back to the slide that I just showed you. Okay? So, polymers. Okay? A junior level chemistry class. This is only a 1-semester class, the idea of how you string together molecules to make polymers. There are a lot of commercially important non-biological polymers. Nylon, I think, being at the top of the list. Rubber has a lot of polymers associated with it as well. So there's some commercially important polymers that people use that don't come from naturally occurring things. But the thing that those non-natural, or those unnatural, I suppose is the best way to phrase it, those unnatural polymers and the ones that our bodies make, that what they have in common is this. That was inartfully said. It really comes down to this. Any polymer, whether it's made in a lab or made by one of our cells, they all conform to this simple rule. What you're doing is you're taking simple subunits and you string them together to make a larger molecule. That's it. That's what's happening with polymerization. When you're making a polymer, you take simple subunits and you string them together. I can dress it up a little bit, and I can tell you you're taking simple subunits of the same kind and string them together. That would be fair. All right? And for each of the 4 biopolymers that we'll talk about, the subunit that you string together is different. Okay, that's why we have 4 different biopolymers. They're each using 4 different kinds of simple subunits. The subunits are often simply referred to as monomers. And when you link one monomer to another monomer, you're beginning to make a polymer. And specifically, if it's only too long, it's a dimer. If you get 3 of them, it's a trimer. If there are 4 of them that get strung together, it's a tetramer, 5, a pentamer. Don't worry about the rest of the Greek, right? But that's what's going on here, is you string together these simple subunits and you make a long chain. In some sense, an infinite chain, a relatively really long chain. Let me give you one particular favorite example of mine. The monomer that gets strung together to make a nucleic acid is called a nucleotide. Okay, those are the monomers that get linked together to make nucleic acids, DNA. Right? Those nucleotides, there are 4 different nucleotides. We're going to talk about that more on Thursday. But there are those nucleotides between friends, right, weigh about 500 Daltons. They have a molecular weight of 500. So all the protons and neutrons added up together for a single nucleotide, ballpark 500 for the atomic weight of all of them. Fancy chemistry talk, that's called a Dalton. There are 500 Daltons in weight. If you wanted to make a 1 molar solution of a nucleotide, here's what you would need to do. You'd need to get about 500 grams of that monomer, 500 grams of that nucleotide. 500 grams is in, roughly the weight of an iPhone, right? About 1/4 pound. You need to take that much of that nucleotide and dissolve it in 1 liter of water, right? Nothing new there. That's just what we've been talking about last week. Concentrations, molarities. Hopefully nothing's different. But try to conceptually picture how you'd do that. You'd take about an iPhone worth of a nucleotide. It's more or less like sugar, white crystalline sort of thing, and dissolve it in a liter of water. A liter of water is like half of a 2-liter bottle of a soft drink, right? Yeah, I can do that. Sure. That should work. The nucleotides are very hydrophilic. They've got some charges associated with them. Yeah, no problem. I can get a 1 molar solution of a nucleotide, no problem. But we don't work with individual nucleotides when we're talking about polymers. We're talking about long chains of nucleotides. The biggest chromosome in our cells, your cells and my cells, human chromosome number 1, has on the order of 200 million nucleotides strung together. Stop and think about what that means. Not just a million, 200 million of these nucleotides all strung together to make a polymer. How much of human chromosome 1 do I need to have in hand if I want to have 1 mole of human chromosome number 1? A nucleotide has a molecular weight of 500, all right? Human chromosome 1, that molecule has a molecular weight of 500 times 200 million. I always hesitate to do math live in circumstances like this, but I'm thinking this is in the ballpark. 500 times 200, that sounds like 10,000 million would be 10 billion? 10 billion pounds. I needed a quarter pound of a nucleotide to dissolve in water. Now I need 10 billion pounds of human chromosome 1. I tell you, as I'm talking about it, that's just too big. That can't be right. Maybe there's been a math error done here. But the point is, I think it's right. To make a 1 molar solution, to have 1 Avogadro's number of human chromosome number 1, I need 10 billion pounds dissolved in a liter of water. 10 billion pounds. Could we even fit 10 billion pounds in this room? Probably not. Maybe. I don't know. It would be an interesting experiment to conduct someday. But it's huge, right? So the point here is really simply this. When you're talking about biopolymers, nucleic acids, proteins, the string of these subunits, the monomers that get strung together, can be mind-boggling compared to the size of the individual subunits by themselves. All right, so human chromosome 1, 200 million nucleotides got strung together. Starch, like you find in pasta, has hundreds or thousands of sugars that got strung together. Right? So that's what's going on with these polymers, is you're stringing together sometimes crazy long chains of the subunits. So that's polymers in general. Specifically, the way those subunits get strung together, and this holds true for all 4 biopolymers, as well as unnatural biopolymers like nylon. The way they get strung together is always, curiously, perhaps, maybe by coincidence, always, though, by what are called condensation reactions. You know what condensation is, right? For those of us wearing glasses, you walk out from you walk into the house from being cold outside, and water condenses on your glasses in a nice warm inside temperature, right? Condensation. That's all we're talking about here. A condensation reaction, see, I got it flipped around. A condensation reaction is how you break these guys apart. The way you link them together is with a dehydration reaction. So in a minute, I'm going to tell you about condensation reactions. Now let's talk about dehydration reactions. If you've got a trimer 3 subunits, and you want to bring in a 4th one to make it a tetramer, all you got to do is take away a water molecule to link them together. So you can see why it's called a dehydration reaction. A water molecule comes out, and you end up making a linkage between the 3rd monomer and the 4th monomer. All right? So dehydration reactions are what we use to string together these monomers. Condensation reactions, darn. Okay, so remember what I said? I made a mistake before about saying a dehydration reaction and a condensation reaction. I was completely wrong. Dehydration reactions and condensation reactions are often called the same thing. They're roughly synonymous. But you can see your book here likes to talk about dehydration reactions. So let's talk about this in terms of what your book says, a dehydration reaction. All right? So you take a water molecule out to link these 2 monomers together and add to the chain. If you want to break this polymer up, you simply do the reverse reaction, the reverse chemical reaction. You have to bring a water molecule in to bust up the linkage between the 2 subunits. So for polymerization, dehydration to extend them, hydrolysis, hydro, water, lysis, breaking, right? Water breaks those bonds to get you back to your individual monomers when you're done with that polymer. Okay? And here are the 2 together. You can see you can recycle these subunits simply by sometimes, well, you recycle by doing a dehydration. Recycle by doing a hydrolysis. And you can reuse those monomers by doing more dehydration reactions, stringing them together. All right, so the 2 go hand in hand. One extends the polymers, the other reduces their size. So we got polymers. Yeah?
On the previous slide, why does it selectively go to one hydroxyl group and not the other?
So are you pointing out that there's a hydroxyl group here as well as here? Chemically, there's nothing different about this hydroxyl group than this hydroxyl group, such that you could probably drive this reaction just as easily from an energetics perspective by linking things on at this end. But I'll tell you this. Practically speaking, the way living things do this, invariably you attach an incoming monomer only to one end. And it's always to the end that has the hydrogen. So the incoming guys always come in like this. And it's rooted ultimately. I don't think it's a chemical difference or a chemical problem. It's a biological thing. There are enzymes that are catalyzing these reactions, and they are geared up to always extend to that one end and never the other end. And when we talk about proteins and nucleic acids, the reason for that fixation on attaching just to one end as opposed to both ends becomes a little bit clearer. I don't know that it applies as well to carbohydrates or lipids, but I can tell you the enzymes that do it for carbohydrates and lipids have the same sort of strange fixation of only adding on to one end and not the other. It's an unsatisfying answer to some extent because what I'm saying is that's just the way it happens, but that's the way it happens. All right, so we've got polymers. Okay, let's talk about what you can do with some of these polymers. Finally, right? Today I want to talk with you about these 2 bad boys here, carbohydrates and lipids. Remember, living things are using these 4 biopolymers to get certain important jobs done. Okay, so one of the things to focus on as I'm talking with you about these biopolymers is what are these molecules being used for? What job needs to be done in order to help something be a living thing? These first 2 have some overlapping responsibilities. Both of them are involved in energy storage. Okay? Proteins and nucleic acids have virtually nothing to do with energy storage. They're doing different jobs. But carbohydrates and lipids both have as their primary responsibility energy storage. In another couple lectures, well, actually, it's probably going to be right after the first test for this class. So in October, I am going to blow you away with how obsessed living things are, particularly bacteria, with keeping track and doing bookkeeping with energy, right? It's a huge deal to living things. Energy, where is it? Where is it being stored? How is it being moved from one place to another? It all revolves around carbohydrates and lipids, right? So that's the primary function for both carbohydrates and lipids. Let's talk specifically about what's going on with carbohydrates. So carbohydrates, the long and the short of it is this. Carbohydrate. Carbo should be conjuring up some thoughts about carbon. These are carbon compounds. And hydrate should be conjuring up some images, some thoughts about water. Carbohydrates all have the same basic chemical formula. For every carbon in the molecule, there is a water molecule associated with that water. There's a water molecule associated with that molecule. So the basic structural chemical formula for any carbohydrate is this. C1, one carbon, for every one carbon, there will be 2 hydrogens and 1 oxygen. CH2O. Depending on the carbohydrate that you're talking about, you may have a different number of carbons. So here are 2 carbohydrates. There's glyceraldehyde and dihydroxyacetone. But check it out. You've got 3 carbons. And so, I'm crossing my fingers here. Let's see how well my prediction holds out. I said we have 3 carbons. That means we should have 3 times the number of hydrogens that you would normally have. There's usually 2, so there should be 6 hydrogens. And there should be 3 oxygens. Oxygens are easy. There's 1, 2, 3. Okay, so my prediction held true there. How many hydrogens? Oh, I don't know. I'm worried about this. 1, 2, 3, 4, 5, 6. Drop the mic, walk away, that's it. I'm not going to be able to do anything better than that for you the rest of the day today. How about that? That's a carbohydrate. 1 carbon. For every carbon, there are 2 oxygen. For every carbon, there are 2 hydrogens and 1 oxygen. Basic structural chemical formula for any carbohydrate. Here, we're looking at some pretty small monomers. These are only 3 carbons that got strung together. But this is a sugar, and this is a sugar, right? Carbohydrates, as a rule, all of them are sugars. Here are some more interesting, perhaps, carbohydrates. 2 more sugars, differing only in terms of where the carbonyl group is attached, but check it out. Ribose is a 5-carbon sugar. 1, 2, 3, 4, 5. Oh, look at that. See, I should have been cheating, right? The answer's right there. I didn't have to get all stressed about what happened on the previous slide. I bet you it was there. Yeah, look at that. It's right there. Oh, well. Here I was worried that I was going to be proven wrong by my own slide, and the slide's given the answer all along. But look at that. 5 carbons, 10 hydrogens, 5 oxygens. I'm remiss in not having challenged you with, when you look at a structure like that, what's it say to you? How's this molecule going to behave? What do you think about a molecule like this? Check it out. Look at ribose. Forget about ribulose for now. Let's just focus our attention over here. What can you tell me about this molecule? Ooh, a lot of hydroxyl groups, right? That's going to want to have some fun party time with water. Water is going to be really interested in hanging out with those hydroxyl groups. What do you think? Might a sugar molecule be water soluble? Right? If I gave you a teaspoon of sugar, would you be worried that you'd have a hard time dissolving it in a cup of coffee? No. Right? That's why. Those hydroxyl groups are going to make this molecule pretty water soluble. The carbonyl group is only going to help make it water soluble. So, at a glance, this should be highly soluble in water. Not hugely chemically reactive, no full charges anywhere, but nonetheless hydrophilic. It wants to hang out with water. What else? Is this going to be the sort of thing that when you put a match to it burns, or will it explode, right? Or will it do nothing? I suppose is my three choices here. How much energy is stored in the covalent bonds of this molecule? For ribose, that hydrogen-oxygen bond is a very polar bond. That's a pretty low energy bond. Low energy, low energy, low energy. Oxygen to carbon, polar bond. Not too much energy there. There, or there. Low energy bond. I'm looking here and I see a whole bunch of relatively low energy bonds associated with this sugar molecule. But that's not the whole story. Because on the other side, look at this. Hydrogen to carbon. Ooh, that's a high energy bond. If I break this bond, a lot of energy is going to come out. Here, 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 here. I got a bunch of high energy bonds. And don't forget carbon to carbon. These are high energy bonds too. So, bond for bond within a sugar molecule, I'm kind of middle of the road, right? Average it all out. I got a bunch of low energy bonds. I got a bunch of high energy bonds. The molecule as a whole, kind of middle of the road. Not bad for energy, right? But you can imagine how you could get it to have more energy than it does. Right? Get rid of some of those hydroxyl groups and replace them with hydrogens, and now we're talking super high energy. But it's not a WIMP like water, where there's virtually no energy in those bonds, because all you've got are polar covalent bonds. Right? So in the continuum of things, it's more energy rich than water, less energy rich than things that you can imagine that don't have the hydroxyl groups on. Right? So kind of middle of the road, energy storage wise, maybe a good idea just to say, oh yeah, there is a fair bit of energy there, but I can imagine how you would do better. Funny thing about chains of carbons in carbohydrates, once the chain gets to be about 5 or longer, 5, 6, 7, once the chain gets to be 5 or longer, in a watery environment, those carbon chains fold back on themselves within the molecule to make a ring structure. And so what's happening is you're simply bending some of these bonds. Here we've got some 6-carbon sugars. This to me looks like glucose. You've got some 6-carbon sugars, and you can fold this back around and pull this up around and then get a covalent bond to be made between the oxygen and the carbon. And you end up making a ring form of that same sugar. So this and this are actually completely equivalent. In a watery environment, this sugar will normally fold up to act like this. And you notice how the chemists here who have given us this structure are kind of taking it easy on us. Over here, they haven't done it yet, but notice how all these carbons are still shown, and they're still shown here. Typically, when you see an angle like this in a structure, particularly a ring structure, you're supposed to just know there's a carbon there. That's a shorthand. But that's a fairly common shorthand structure that's equivalent to this structure here. So a lot of times when you see these ring structures, nobody's going to draw attention to the fact there's a carbon there. They're still there. So there you go. You got these abbreviated ring structures. And where's the polymer part of all this? Well, let's do a dehydration between one glucose and another glucose. One monomer plus another monomer. We got a dimer. Dehydration is going to come into play to pull that off. And look at that. We can take one glucose, hook it up to another glucose, and get a whole different named sugar. That disaccharide of glucose is called maltose. This is a very stable relationship. These are all covalent bonds. They're not going to fall apart unless something comes and gives them a little bit of a push. We had 2 monomers with a certain amount of energy associated with it. We now have a dimer with twice as much energy associated with it. And the bond that links the one monomer to another monomer gets a fancy name. For sugars, that's called a glycosidic linkage. Sorry, that's just the way it is. That's the name of the bond that links one sugar monomer to another, glycosidic linkage. You don't always have to link glucose to glucose. You can make other kinds of disaccharides. You could take a glucose and hook it up with another sugar. This one is very similar to glucose, but a little bit structurally different. Notice how the hydroxyl group is pointing down here. It's pointing up over here. Right? So there's some subtle differences, but if you link up glucose with fructose, you get a different sugar. That's called sucrose. And again, you've still got that glycosidic linkage. You don't have to stop at dimers. You can make trimers. You can make millionmers, whatever the right Latin word for million would be. I don't know that they went that high. But you can make long, long polymers. And it's convenient for biologists to break those polymers up into different categories. If it's just a long chain of monomers linked together, that's usually referred to as amylose. If it's a long chain that once in a rare while has a branch, so in addition to the 1,2 linkage, it'll have a 3,5 glycosidic linkage, somewhat unusual. But if there are some chains, that gets called amylopectin. And if there are a lot of chains, it gets called glycogen. If an individual monomer, each of these little green symbols here, if an individual monomer is a sugar, and if a sugar has a reasonable amount of energy associated with it, a long chain of them is going to have more energy still, right? You don't need me to tell you that, but that's just the way it is. So if you link together a bunch of glucose molecules, I've just said you could call them amylose, you could call it amylopectin, you might call it glycogen. But when you zoom in at the spots that don't have branches going on, what you would get is what you see at the top of the slide there. These are all starches, long chains of monosaccharides together. These are all carbohydrates. Each of the individual monomers is a carbohydrate. Strung together, it's still a carbohydrate, but a long chain of them, amylose, amylopectin, and glycogen, all of those are different kinds of starch. Right? So starch is something that's a long chain of sugar monomers linked together. And what do you associate with starch? Right? Pasta, perhaps the most commonly encountered starch in our diets. Right? Pasta, long chains of sugars linked together. It's not that different. When you eat a bowl of pasta, aside from the taste and the texture at a chemical level, it's not that different than eating a bowl of sugar. The only thing that's different is they're strung together. Calorie content, no difference between a bowl of pasta and a bowl of sugar. None. It takes a little longer for us to break apart the starch to get it into its individual component monomers, these individual sugars, and so it takes a little while longer for you to get that sugar rush, but you still get the same amount of energy, and it's because ultimately, structurally, chemically, they're the same thing. So that's how things get strung together to store energy. Starch is a great molecule for storing some energy, for moving energy from one place in a cell or one part of an organism to another part of an organism. It's water soluble. Right? Nice, convenient, handy that way. The individual monomers are sort of like the $20 bill of the energy currency for cells. There's a good chunk of change there, so an individual monomer is nice. A long chain of them is better still. Right? Got money in the bank, but they're water soluble. They're good for storing energy. They're good because of their water solubility. They're good for moving that energy. Sometimes in smaller chunks, sometimes in big, massive, armored car kind of like chunks from one part of the cell to another. If you link these monomers together just a little bit differently, so instead of having them line up in this fashion, you sort of flip them around in their glycosidic bond and lock them into place that way, you make a carbohydrate that has some very different chemical activity, very different chemical behavior. I'm pointing now to cellulose. Cellulose, like the paper that many of you are writing on, is mostly cellulose. Cellulose is nothing but a carbohydrate. And there's this fair bit of energy stored in cellulose. That's why we burn wood in fireplaces to keep warm in the winter. And that's why if you put a match to a piece of paper, it will burn and give off heat, because what you're doing is you're breaking those bonds in the individual monomers, the individual sugars that got strung together. You're combining oxygen with the carbohydrate, and you're releasing carbon dioxide gas and water. End result of that burn that happens. So you could burn glucose if you like, but trees don't have big, long chunks of glucose or starches for you to burn. Instead, they've set things up differently for us as cellulose. Cellulose isn't used by plants for the purpose of energy storage so much as for structural purposes, because of the rigidity of that molecule compared to what you get in a starch. So we had a biopolymer, pretty good at storing energy. Apparently, at some point during the course of evolution, it got co-opted. This variant got used also to provide a structural support, as well as a repository for energy. I have it in the notes. I think this is a curious bit of trivia for you. But many people will tell you that the single most abundant biopolymer on the planet is cellulose. Right, so this is the all-time winner in that regard. And it's a cool picture, right? It turns out also that there are other things you can do with carbohydrate polymers, right? Use them for storing energy. You can use them to make cellulose for structural purposes. Chitin is just a subtle variant on cellulose. So the exoskeleton for insects is made out of chitin, and it too, chitin, is also a biopolymer. Here's glucose. A little while ago I showed you what cellulose looked like. Not that much difference in terms of what chitin looks like. You can see it's a carbohydrate, it's similar, but there are some subtle differences. Those subtle differences equate then to the difference between starch and chitin. You want to burn some exoskeletons? They burn really well. Just put a match to them.
Well, would you say that's the reason why people like to eat bugs in the wild?
I don't know why people eat bugs. I can't get inside of their heads, but I can tell you that those people who do eat bugs are getting a pretty good carbohydrate from chewing on the chitin. There's probably some good energy there that they can metabolize from them. So there you go. That's carbohydrates. Boil it all down. What are we talking about for carbohydrates? Carbohydrates, the individual monomers are sugars, right? Monosaccharides, mono one saccharide sugar, right? If you link together multiple of those monomers, multiple subunits of those sugars, you do it by glycosidic linkages. You can use those glycosidic linkages to string together long chains of these sugars to make starches that are great for energy storage as well as energy delivery because of their water solubility. Variants on the theme can also be used for structural purposes. But when we talk about carbohydrates, for the most part, our eye is going to be on the ball of energy storage or energy transfer. Let's move on to perhaps the most boring. That's just what you were hoping to hear, right? Let's talk about the most boring of the biopolymers, the lipids. Okay? You guys probably don't know much about Monty Python and the Flying Circus, but I'm tempted to launch into something about the larch. So for those few of you who know about that, you might find that interesting. But lipids. Nobody finds lipids exciting, all right? So let's rip it off like a Band-Aid and move on to something that might be a little bit better, right? what's up with lipids? Here's what's up with lipids. They're a biopolymer. And just like carbohydrates, their primary purpose is energy storage. Unlike carbohydrates, where energy storage and water solubility and delivery came into play at the same time, lipids are not water soluble. They're hydrophobic. And so while they're great at storing energy, not so good at getting it from one part of a cell to another, and also not that good in terms of giving that energy up once you've put it in. It's sort of like the difference between having a bank account where you can just go and withdraw the energy whenever you like, that's the carbohydrate in this analogy, and a certificate of deposit where you enter into a relationship with the bank and you say, I'm going to give you my money and I'm not going to ask for it back in really big chunks unless I give you a lot of notice. Right? There's certain restrictions on how I can draw the money out with a certificate of deposit that don't apply for when you've got just a regular old-fashioned bank account. There's advantages to both, right? So seek out a qualified financial advisor to guide you with your own personal circumstances. But that's the difference. Carbohydrates, you want energy, I'll get it to you right away. You ask, it's there right away, whenever you need it, and I can get it from one place to another very easily. Lipids, lots of energy, not bad in terms of the energy stored, but it's a little harder to crack into it and get that energy out when you need it. That doesn't mean you can't, it's just you have to go through a few more hoops. So, for lipids, it's a biopolymer. The monomers for lipids are fatty acids. And unlike the other biopolymers, you don't usually link together huge numbers of them. You'll typically, for most living things, link together just 3. So you'll take 3 fatty acids and hook each of them to a single glycerol to make this biopolymer, which is a fat molecule. Okay? Glycerol is a 3-carbon carbohydrate. 3 carbons, 6 hydrogens, 3 oxygens. And the fatty acid, aside from that one little carbolic acid way over here at the end, this chain, 16 or 17 carbons long for a typical fatty acid, this chain is nothing but carbon to carbon and carbon to hydrogen bonds. You hook up this fatty acid to the glycerol through a dehydration reaction. So just like the other polymers, you're going to kick out a water molecule. And when you're all done, you're going to make these things called ester linkages. Not glycosidic linkages here, but rather an ester linkage. And you end up with a fat molecule that looks like this. It's a triacyl glycerol. 3 of these acyl groups get hooked together to this glycerol to make that fat. So let's do the sort of standard thing. When I show you a chemical structure like that, I hope you're way ahead of me at this point, and you've already figured it all out. When you look at that molecule, what are you thinking? I'll tell you what I'm thinking. I look at that triacyl glycerol, and you know what I'm seeing? I'm seeing really very little that could form the basis of a meaningful relationship with water. All right? The ester linkage maybe has some water solubility associated with it. But I tell you, as I look at that, I'm thinking that little bit of water solubility from the ester linkage is going to be so swamped out by what's going So I think what we've seen happen here is the battery has run out for my cordless microphone, and I'll have to be tethered. But that's probably best for you. I'm a little safer when I'm anchored down to the podium. So yeah, check it out. This is not going to be very water soluble, all right? But what else is happening here? What else is in play? You know what else is in play? Not only is it not very water soluble, but that thing, that triacylglycerol, bond for bond, check that out. Could you design a molecule that has more high energy bonds in it? Maybe you'd have to do something with those ester linkages, but short of that, man, every bond there is a nonpolar covalent bond. That is pegging out the energy richness quadrant for us when we're talking about fatty acids and lipids here, right? So, lipids. First thing that should probably come to mind, wow, they've got a lot of energy associated with them. And ooh, boy, I want to talk about something that's not water soluble. There's the gotta be the all-time champ of not water soluble. All right? So that's what's up with lipids. You may have heard when you're talking about fats, about saturated fats versus unsaturated fats. This is a good time for me to delve into that topic with you. For a saturated fat, it boils down simply to this. See how they've tried to not confuse you with all the carbons and hydrogens? They've just shown you this squiggly line. Each of the angles is a carbon, and coming off of each of those angles are two hydrogens. Right? So here it is in all its glory. Here it is in the shorthand. Bottom line, for a saturated fat, super easy to remember. It's saturated with hydrogens. Every place you could dangle a hydrogen off of a carbon, you dangle a hydrogen off of a carbon. That's what makes it saturated. It's saturated for hydrogens. An unsaturated fat differs in that, at least in a few places, in place of hydrogens, you form another bond between the two carbons. So there's a double bond here between these two carbons. And to make that double bond and keep the valences happy, you had to kick out two hydrogens. So the unsaturated fat is missing a couple of hydrogens compared to the saturated fat. Let me challenge you with a question. Which of these two structures has more energy stored in its covalent bonds? This one or this one? It's kind of a trick question because the answer is they're the same. The energy stored in a carbon-carbon bond is the same as the energy stored in a carbon-hydrogen bond, right? So there's no difference in the bond-for-bond amount of energy stored in an unsaturated fat versus a saturated fat. The difference is a structural one. You can see that double bond introduces a kink, a bend in that fatty acid chain. Okay, whereas here it's a nice long straight wiggly thing that can bop around. That double bond puts a bend in the chain, and it also locks that into place to some extent. And what ends up happening at a molecular level is that you can stack these guys, saturated fats, one molecule on top of another very regularly, much like you can lock in water molecules in ice. You know, that regular structure is something that happens for solids, right? So saturated fats at room temperature tend to be solids. Unsaturated fats at room temperature tend to be liquids. Butter versus oil. All right. So, zoomed in picture. Couple more things. I talked with you about cellulose and chitin and how they got co-opted in the carbohydrate game to not just be things that stored some energy. Actually, now their primary purpose is more of a structural one. Well, with lipids, the same sort of thing has played out. Lipids primarily seem to have been around to be long-term energy storage molecules. But if you replace one of the triacylglycerides, if you replace one of the acyl groups with a choline, you still keep your glycerol and you keep your two other fatty acids, you get what we call a phosphodiester you can make a phospholipid because of this phosphate linkage here, right? So phospholipids are a subtle variation on the theme of the triacylglycerols. Here there's only 2 acyl groups and a phosphate group with a choline attached to it. It gives you a structure that looks like this. I tell you what, when you see a structure like this, let's talk about this. What's going on with that structure? How's it going to behave? Long and the short of it, this is a messed up molecule. It's literally schizophrenic. It's literally got 2 completely different kinds of behavior. At this end of the molecule, this is totally hydrophobic, wants nothing to do with water. At this end of the molecule, because of that phosphate group with its 2 negative charges, as water-friendly as you could ever find, right? You're at both ends of the spectrum. In one molecule, I told you there's this continuum of hydrophobicity versus hydrophilicity. This one molecule does it both. It's part of it resides at this end of the spectrum and part of it resides at this end of the spectrum. This part of the molecule is going to be very water-soluble, this part very insoluble. Let them hang out together, right? One of these molecules with others, and it naturally forms into a situation like this, where the hydrophobic tails rub up against each other and exclude water, and the hydrophilic heads all line up on the other surface so that they can interact more with water. This is a phospholipid bilayer. And we'll talk about that more in the context of membrane structures. So let me leave you with a question to ponder between now and the start of class on Thursday. Which of these 5 different kinds of things, 1-pound quantities of each, has the most energy associated with it? If you prefer, which one of these will have the most calories associated with it? 1 pound of each of them. I give you the choice of sugar, pasta, vegetable oil, butter, or water. All right? Should be pretty obvious after what we've talked about today, but we'll leave it at that, and I'll see you guys on Thursday.